Hello, everyone. Let's dive into mitosis. All right, let's get this shrunk down. Please excuse my dog who's probably about to bark. All right, so we have two goals for cell division. The first is growth, maintenance, and repair of cells. The second is reproduction. Mitosis accomplishes both. Now, we've talked a lot about DNA so far, and in eukaryotes, we have 23 pairs of chromosomes that encode and contain our DNA. We have what are called 22 somatic pairs of chromosomes, or autosomes, and then we have one pair of sex chromosomes down here. Okay. Oops, sorry, there's one and there's one. So within species, chromosome number is usually, whoopsie daisies, usually pretty consistent, but can vary with the different developmental stages or specific cell types. In our case, we're going to talk about different cell types. So what I mentioned before were those somatic cells. Okay, in biology, soma means body. So somatic cells are body cells. Okay, and gametes are sex cells. Okay, so somatic cells typically have two matched sets of chromosomes, which makes them diploid or 2N. These are important terms to remember, especially when it comes time to compare mitosis and meiosis. Gametes are egg cells and sperm cells, and they only have half the number of chromosomes. Why would a cell only have half the number of chromosomes? Well, eggs and sperm have some specific jobs, right? Their jobs are to come together to form a zygote. A zygote is actually 2N, okay? So our eggs and sperm are haploid or 1N, and haplo means single, okay? So, or half, single or half. So eggs and sperm only have half the amount of chromosomes or a single chromosome compared to somatic or body cells, which have two copies, two matched sets of chromosomes, making them diploid. Di means two. So what we just looked at was a karyotype. That's this here, a layout of all the chromosomes in the body. Okay, and again, we have that X and Y pair of chromosomes. Okay, most chromosomes have that pair, that are in that pair, have what are called homologous chromosomes, meaning that they are identical. Homo means same. Sorry, that didn't show up. Same. Some genomes, like mammals and birds, for example, in mammals, our sex chromosomes are X and Y, and in birds, for example, they are Z and W. These are heterologous pairs of chromosomes, or different, meaning they contain different types and amounts of DNA, whereas homologous chromosomes contain the same DNA in the same spot, not necessarily the exact same DNA, but on the same spot, and are identical. So, organization of a chromosome. So at its largest, okay, we have a duplicated chromosome. So we have one chromosome here and one chromosome here, and they are joined by at the centromere. This is the centromere. Okay, if we unwind that, okay, we have what we find as and refer to as chromatin. And then if we further unwind that, we have what are called nucleosomes that are coiled into chromatin fiber. sorry, we're building. I started here. And then if we continue to unwrap that, we find DNA wrapped around histone proteins. And then if we continue to unwrap that, then we find our double helix with our A's, T's, C's, and G's. So we're not going to talk about this packaging of DNA. It's there for your info. The cell cycle, okay, is the life cycle of a cell, and mitosis is part of that life cycle. Now, I'm not going to go in detail about the cell cycle here, but you have those slides to review and those chapters to read in your book. So mitosis itself consists of I don't include prometaphase, so I usually just talk about prophase, metaphase, anaphase, telophase, and cytokinesis. Okay, these are all the stages contained within mitosis, which is the reproductive part of a cell's life if you are a body cell. Okay, so during prophase, our chromosomes condense and become visible. Okay, so they go from being in those unwound and in the double helix form to being condensed, wrapped around those balls of histone protein and condensed into chromosomes. The nuclear envelope is going to break down and the nucleolus is going to disappear. Then during metaphase, we are going to have what are called spindle fibers develop, and centrosomes located at the opposite of each side of each cell, or the cell. And then in the middle here, you have all the chromosomes lined up. Meta specifically means middle. Okay, so in metaphase, the chromosomes line up at the middle of the cell, and in prophase, pro means first, so this is our first stage. Anaphase is up next, and ana means up. Okay, so our chromosomes are actually our sister chromatids, because there's two of them, are going to be pulled apart at that centromere in the middle and pulled up to these opposite ends of the cell. That's why it's called anaphase. Telo means final or last. So this is the last official phase of mitosis, but cytokinesis still counts, where basically the events of prophase are reversed. So the nuclear envelope begins to reform, the mitotic spindle is going to break down, chromosomes are going to begin to decondense and unwind. Then we have cytokinesis, which is where the two cells physically separate from one another. This happens differently in animal cells versus plant cells, whereas in animal cells, what's a muscular contractile ring called a cleavage furrow forms, whereas in plant cells, a cell plate forms and separates the two cell walls. And they're different because animals don't have cell walls and plants do. Okay, so we talked about prophase. Metaphase, chromosomes lining up in the middle. Again, these sister chromatids, remember, we have sister chromatid 1, sister chromatid 2, joined in the middle by that centromere. Okay, so our sister chromatids line up in the middle, metaphase, of the cell. And then at anaphase, ana means up, our chromosomes are pulled up towards the poles of the cell, and the cell elongates. During telophase, the events of prophase are reversed, so chromosomes are going to unravel, the nuclear envelope is going to reform, and your centrosome and spindle fibers are going to break down. During cytokinesis, in animal cells, you have the formation of the cleavage furrow, which is a contractile ring that helps to basically pinch off the two cells into two separate cells. And in plant cells, you have the cell plate because of the fact that you have cell walls, and then that is what differentiates between the two cells. So during the cell cycle, we have regulation at certain points, and this is where I'm going to talk about the cell cycle a little bit. Okay, so G1 is our cell growth phase, S is synthesis, and G2 is another growth phase before, but it's a specific growth phase before mitosis. Okay, and there are checkpoints before you enter into each next stage of the cell cycle to make sure that your chromosomes are duplicated properly and that everything is going the way it should go to have proper cell division happen. So we have a checkpoint near the end of G1 right here. We have a checkpoint at the end of G2, and then we have a checkpoint in the middle of metaphase during mitosis. Okay, control of the cell cycle is really important, and external triggers can actually initiate or inhibit the cell cycle, and this can cause cancer. So these checkpoints, especially the G2 checkpoint, is the most important checkpoint because this ensures that chromosomes have been replicated and that the replicated DNA is not damaged. If DNA is damaged, this can cause out of control cell reproduction, or what's called proliferation, which is the accumulation of cells in one area, which causes a tumor. Okay, so cancer covers many different diseases that are characterized by uncontrolled cell growth. This begins with a gene mutation that results in a faulty protein regulating cell reproduction. Okay, so tumors can reproduce at higher rates than normal cells, and that's also why they're a problem. There's something called proto-oncogenes that are normal genes that code for positive cell regulators. When these genes, one example is this P53, okay? When these genes mutate, they become what we call oncogenes, all right? Because they're proto-oncogenes because their disruption is what leads to cancer. Okay, so they're potentially proto first, preliminary, they're potentially cancer-causing genes. The reason that is that the gene codes for a protein. Gene codes for protein. Oh no! I learned how to erase. I have an eraser now. But I have to get it back, or I have to get my pen back, and we'll change colors. I always forget to do that. The gene codes for a protein. If there is a mutation in the DNA, it means there is a mistake in the gene, and it means that that gene will produce a protein that is damaged, okay? So if you have a mutated P53, meaning your protein is a different shape, okay, you can have the cell cycle continuing even with damaged DNA, and you can end up with cancer. Okay. There are certain what are called tumor suppressor genes that are segment of the DNA code that help to regulate. There are negative regulator for such proteins. So when these are activated appropriately, they can prevent uncontrolled division, but sometimes those regulators can get mutations, and then those can also cause cancer as well. Don't need to keep the ink annotations. So when we get to meiosis, okay, mitosis and meiosis are incredibly similar with the exception of a few certain events that give us genetic variation, and then there are some functional differences that you're going to need to know for your exam that we will talk about. Have a wonderful day.